Hello, students. Hello. Are you able to hear me? Hello. 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 Hello, students. Are you able to hear me? Hello. Hello.
Yes, ma'am.
No one is responding. Why?
Good afternoon, ma'am.
Good afternoon. Good afternoon. Okay.
Thank you.
So, yeah, now I think almost about 6 to 7 students have already come. So, we will start the class. As you all know, I am going to deal with course 2, that is genetics and evolutionary biology. So today we will be discussing on the topic gene. What is gene and what are the different types of gene? That is the structure of gene. What we call it as a concept of gene. Okay. So you all know actually gene is the fundamental unit of heredity. Okay. So it is actually a molecular unit of heredity, we can say, or a fundamental unit of heredity. So almost all the living beings actually depend on genes because what do? They specify all the proteins and also the RNA chains. So they hold the information to build up and also maintain an organism cells. And the genes actually, you all know, are passed from one generation to another generation. That is, they are passed from the parents to the offspring. So gene actually serves as a very, very important purpose. So that is why it has been regarded as a fundamental unit or a molecular unit of heredity of almost all the living organisms. So actually it is transferred from a parent to the offspring, and because of the gene actually helps in determining the characteristics of the proteins. So gene, where is gene located? You all know actually gene is present on the chromosome at a specific point, which we call it as nothing but the locus. So a gene is a locatable region of the genomic sequence, which corresponds to the unit of inheritance. And it is associated with a regulatory gene, transcribed genes, and also other different functional sequence regions. So when coming to the history of the gene, you all know actually the existence of the genes was first being suggested by Gregor Mendel. You all know Gregor Mendel was the father of genetics. So he tried to actually study the inheritance of these genes using the pea plant. And what he did is actually when he was studying the pea plant and studied the inheritance in the pea plant, he hypothesized or he gave one factor, or he said that if a factor is there which is responsible for conveying the trait from the parent to the offspring. Later on, another scientist by name Danish botanist Wilhelm Johans, actually he coined the term or the word gene in the year 1909 to describe this fundamental physical as well as a functional unit of heredity. So we can say the genetic word was first used by William Bateson. So like this, when we talk about the history of the gene, three authors actually are associated with this gene. One is the existence of the gene was suggested by Mendel. Then later on, the Danish botanist by name Wilhelm Johans, he coined the word gene, and later on, the genetic word that is the gene was first used by William Bateson. So this is about the history of the gene. So what is gene? Gene is nothing but a molecular as well as a fundamental unit of heredity of almost all the living organisms. All the living organisms actually we depend on genes because what do? They specify the proteins, they specify the RNA chains, and they are being transferred from the parent to the offspring. So whatever the genes we are having, actually we have been, it has been transferred from our parents to us. So like this, the chain will go on. Okay, so it is actually being present on the chromosome at a specific point, which is called as the locus. Now, next is Darwin. Now, the scientist by name Darwin, you all know actually, who is the father of evolution. He used the term gene mule to describe a microscopic unit of inheritance. And later on, this microscopic unit of inheritance later on actually became known as the chromosome. which has been observed, which actually is being observed during the cell division. Then Morgan came. Morgan became the first, actually began the first chromosomal map of the fruit fly, that is Drosophila. Then next, James Watson and Francis Crick, you all know, they gave the structure of the DNA. And later on, Walter Fiers and his team at the Laboratory of Molecular Biology were the first to determine the sequence of the genes. So this is a brief history of the gene. So you all know actually the term gene was first coined by actually Gregor Mendel. He used the term gene. So gene actually is a what a fundamental unit of heredity, or it is a fundamental unit of heredity.
Heredity.
Yeah, 1 second, sorry guys. So it is actually the existence of the gene was first suggested by Mendel. Later on, Wilhelm Johans coined the term gene, and later on it was first used by William Bateson. Now coming to next, that is a Darwin. He used the term gene, okay, or you can say the gene mule to describe a microscopic inheritance. Morgan actually gave the first chromosomal map using the fruit fly, that is Drosophila. James Watson and Francis Crick, you all know, actually they have given us the molecular structure of the gene, and Walter and his team actually gave us, or they were the first to determine the gene sequence. Now coming to the structure of the gene. So we all know actually what are genes we know. Now they are present on the chromosome at a specific point called as the locus. When we talk about the structure of the genes are present actually in the form of a granular structure, okay, on the chromonemata of the chromosome. You all know actually what is chromonemata. Actually before the cell division, you all know chromosomes are present in the nucleus. So before the cell division starts, these chromosomes actually they form a network like structure, and they are coiled around, which we call it as nothing but the chromonemata. Only during the time of cell division, or the time of cell division only, this network will break, and they will actually try to gain the shape. So there actually during the metaphase, you all know, we will be able to see the structure of the chromosome. But before that, the structure of the chromosome actually is irregular in the form of a network. That network we call it as nothing but the chromonemata. And it is actually in the form of a granular structure. So the model of the gene structure actually was given or proposed by Benzer, and that is the reason why it is also called as the Benzer model. So according to his concept, what he said is the gene structure, or the structure of the gene, you can say, or the gene has got three units. What are those? One is called as the recon, second is the muton, and the third one is the cistron. So these are actually the three, what we can say, the units which are present in a gene. Now coming to the recon. So what do you mean by recon? Recon is nothing but a unit of recombination. Now what do you mean by recombination? Recombination is nothing but combining of the two genes. Okay, so this smallest unit of recombination which occurs during the crossing over. Okay, so when actually the genes cross over, during the recombination phenomena, you all know actually the genes are being exchanged from the parents to the offspring. So that is what we call it as nothing but a recombination. Recombination is nothing but the crossing over. So during this particular phenomena, we observe that recon. So what is recon? Recon is nothing but the unit of recombination, and it is a very very smallest unit actually which is present on the chromosome, and this is actually helps in the crossing over. Now next is what? The muton. Now muton actually is the unit of mutation. Now muton is the unit of mutation, means what? It has the capacity to undergo mutation, and it also consists of a single base pair. So but at practical, you all know actually each base pair consists of several other nucleotides may map at the same size. So what we can say in simple words is muton is nothing but it is a unit of mutation which has got the capacity to undergo mutation. So what is recon? Recon actually is a unit of recombination. What is muton? Muton is actually the unit of mutation which has got the capacity to undergo mutation. Now next is the cistron. Cistron actually is nothing but the functional unit of expression. This is the last one. So actually what it has now, the genes actually they try to express the characters. They try to express the hereditary characters. So cistron actually is responsible for that. So that is why it is called as the functional unit of expression. So cistron actually is responsible for the synthesis of the polypeptide gene. or polypeptide chains. So the number of nucleotides actually is a cystron, and it may vary between 100 to 30,000 actually the nucleotides. So if you take a gene, the gene has got a number of nucleotide pairs will be there. So this varies from 100 to about 30,000. So if a gene actually is made up of more than one cystron, then what happens? It is able to synthesize more than one polypeptide. So that means what we expect is the gene should actually consist of more than one cystron. If it has more than one cystron, then it will be able to synthesize more than one polypeptide. So the number of nucleotide pairs that are actually present in the gene actually ranges from 100 to 30,000. So this is about the fine structure of the gene, you can say, or the concept of gene, you can say. So what we can say is the gene actually consists of three, what is a regions. What are those? One is called as the recon, muton, as well as the cystron. Recon is the unit of recombination, which helps in the crossing over phenomena. Muton actually is the unit of mutation, which has the capacity to undergo mutation. And cystron actually has got the capacity to express itself. So how it can express? Because it has got the capacity, so it can synthesize a polypeptide gene. So the number of nucleotides varies from 100 to 30,000. So if a gene consists of more than one cystron, then that cystron actually will be able to synthesize more than one polypeptide. So this is the basic structure of the gene. Now where they have talked about the exon, intron, as well as exon. Now I'll come to that later. So you all know what is the concept of gene or the structure of the gene, a brief history of the gene. Is it okay, students? Hello? Are you able to follow?
Yes, ma'am.
So this is how you have to talk. So first, what is gene? A brief history of the gene. So gene is nothing but a functional unit of heredity. It is an important aspect of all living organisms because the gene actually will specify the proteins as well as the synthesize the RNA. So because of the gene, actually the genes are being transferred. Because why we are calling it as unit of heredity? Because these factors have been transferred from the parent to the offspring. So they carry the information. So they whatever the genes that the parents possess, they are being transferred, okay, to the offsprings. And coming to the history, already I have told you, so many scientists actually have been involved with reference to the history of the gene. Like you have Gregor Mendel, then Bateson is there, Charles Darwin is there. So every actually scientist in their own way, they try to actually define the gene, they try to characterize the gene, they can give try to actually give the structure of the gene. So that is why today we are able to know actually what is gene, what is its structure, and all those things. Okay. And nothing the next aspect is your concept of gene, or you can say fine structure of gene. So according to the fine structure of the gene, any gene, if you take for example, consists of three regions. What are those? Muton, recon, as well as a cystron. Now next is what? Now the other actually terminologies that are related with this. Apart from these, actually these are the three basic what do you say, the regions that are being present in a gene. Now apart from these, you also have the other terms which are actually referred. Now one is called as the complon, operon, and the replicon. Now what do we mean by complon? Complon is nothing but a complementary unit of the cystron. It is a complementary unit of the cystron. So a pair of genes having mutation at different cystron or complons will show the complementation. So this will result in the formation of a normal protein in spite of a mutation. So are you able to understand what is complon? It is nothing but a complementary unit of cystron. So a pair of genes having the mutation at different cystron or complons will show the complementation phenomena. As a result, what happens? It will result in the formation of a normal protein instead of a mutation. Next is what? Operon. Now, operon actually is nothing but a set of genes. It is a set of genes which actually in a coordinated manner will control or regulate the expression of the structural genes. So you all know actually the genes are of different types. You have structural genes are there, soluble, different types. So structural genes actually means nothing but it will help in maintaining the structure of that particular gene. So operon actually is involved in this. So operon actually is nothing but a group of genes or a set of genes. So what it will try to do? It will try to control or regulate the expression of the structural genes. Now the operon concept you might be knowing. So it is being regulated, and first of all it is being proposed by Jacob and Monod. So according to this, you have got different types. So you need operator gene, you need the regular genes, and you need the structural genes. So when you talk about an operon concept, okay, in the operon concept actually, which is being proposed by Jacob and Monod, the gene regulation is being controlled or it is being operated by three different types of the genes. One is called as operator gene, regulator gene, as well as the structural gene. So operator and the regulator genes will switch off the expression of the structural genes. So like this, so we should know what is complan, what is operon. Now next is the replicon. Now replicon actually is a unit of DNA replication. Now before we talked about recon, so don't get confused. Replicon is different, recon is different. Okay, so it is a unit of DNA replication. So a chromosome may have a number of replicons will be there. So these are actually apart from the three concept, the regions, okay, with reference to the concept of gene or the structure of gene, that is muton, recon, and the cistron. Okay, the other terminologies in relation to the structural gene are what? Nothing but the complan, operon, as well as the replicons. Actually, these will be helpful. Only operon actually helps in the operon concept, and whereas the complan is a complementary unit of cistone, which actually helps in the formation of normal protein, whereas replicon actually helps in the replication phenomena. Okay, so this is all about the the other terminologies or the other terms apart from the common three regions that actually a gene possesses. Now coming to the types of gene. So before you go to what is actually the different aspects of the gene and all those things, we have seen the structure of a gene. Now coming to the types of a gene. So basically there are three different types of gene. One is called as a prokaryotic gene, eukaryotic gene, and the viral gene. Means the gene which is present in prokaryotes, you say it is a prokaryotic gene. Gene present in a eukaryote, you say it is a eukaryotic gene. And gene that is being possessed by the viruses, you call it as a viral gene. You all know. So prokaryotic cells, you all know, like bacteria and all those things, they are usually found grouped tighter in the operons. So there are many types of these prokaryotic genes are there. So prokaryotic genes are what? Nothing but example, best example is you can give a bacteria, and they are actually found, okay, in groups called as nothing but the operons. And you have a number of prokaryotic genes are there. So what are all those things? We will see. Okay, now one is called as a regulator gene, then promoter gene, and the operator gene. Now this is, I'm talking with reference to the prokaryotic gene. You all know types of genes when we're talking about. Three different types are there. One is called as a prokaryotic, second one is eukaryotic, and the third one is a viral. So with reference to prokaryotic, best example you can take it as bacteria. So in the bacteria, if you take actually this prokaryotic gene actually is present in a group. They work together. It not a single gene will work, but a group of genes actually will work together. So what are all those things? Genes, they consist of a regulator gene, promoter gene, as well as a operator gene. Now coming to the regulator gene. Now the regulator gene actually is responsible for the production of a protein called as a repressor. And this repressor actually may be active or inactive. The active repressor is also known as aporepressor. So this regulator gene actually has got the affinity for the operator site of the operon and can build up with the operator gene, disrupting the movement of RNA polymerase during transcription. So in this way, what happens? Due to the presence of the active repressor, production of messenger RNA will not take place. So thus proteins or the enzymes are also not formed. So that is actually, we are talking with reference to the lac operon. Okay, you know what is operon concept. So operon concept actually needs three different types of genes. One is regulator, promoter, as well as the operator gene. So the regulator gene is responsible for production of a protein called repressor. And this repressor may be active or inactive. And if this active repressor actually has got the affinity, okay, to bind with the operator gene. So what it will do when it binds with the operator gene? It will disrupt the movement of RNA polymerase. So as a result, what happens? Because of the presence of this active repressor, messenger RNA will not be produced. So the protein, so when the messenger RNA is not produced, definitely the enzymes or the proteins also will not be formed. Now coming to the promoter gene actually is located in between the regulator and the operator genes. And it provides the site of attachment, or you can say the attachment site or RNA polymerase during transcription of messenger RNA. So it will provide the site for attachment of RNA polymerase. So thus it decides the activity of the enzyme. So it will promote, whereas regulator gene will not promote, but promoter gene will try to promote, and it will provide a site for the attachment of the RNA polymerase. But in this case, what is happening in regulator gene? It will disrupt the movement of the RNA polymerase, thus inhibiting the formation of what? The formation of the proteins or the enzymes, or production of first messenger RNA. Obviously, when messenger RNA is not being produced, definitely the proteins or the enzymes also will not be formed. Coming to promoter, so it is between the regulator as well as operator gene. So its role is what? It will provide the site for attachment of the RNA polymerase so that it undergoes transcription. So when once it undergoes transcription, then the activity of the enzyme actually will go on. Now coming to the operator gene actually is present between promoter and the structural genes. So it operates the movement of RNA polymerase along the DNA template. So attachment of this enzyme at the promoter, what it will try to do? It will try to interact between the operator and the repressor protein. So it is actually a decisive factor for controlling the action of the RNA polymerase. So it will try to operate or it will try to control the action of the RNA polymerase. Yes, are you all following, students? Hello.
Yes,
Yes.
ma'am.
Okay. Okay, so this is about the structure of the prokaryotic gene. Just now I have told you, no. Okay, so this is the operator, promoter for lac operon, structural gene. Okay, so this is the promoter for regulatory gene. This is a regulatory gene, and this is the promoter, this is the operator. Okay, and this is the rest of the thing actually is a structural gene. So actually this is an example of a lac operon. So what we can say is actually the in case of the what is that? A prokaryotic gene. Prokaryotic gene are of three types. One is called as a regulator gene, the operator gene, as well as a promoter gene. Okay. Now coming to the eukaryotic gene. Now eukaryotic gene, you all know actually that is present in all eukaryotic organisms. So this what is the main role of this is actually this eukaryotic zone when compared to the prokaryotic genes. Actually, the eukaryotic gene structure is highly complex. Okay, particularly in the higher eukaryotes. So this often actually involves a number of discontinuous segments are there that must be assembled into the final messenger RNA. So when you talk about, so actually basically speaking, prokaryotic gene, if you know, it is nothing but it is an entangled mass, or you can say a network of the chromosome, that we call it as nothing but the chromonemata. But in case of eukaryotic gene, it is highly complex, where it has got a number of discontinuous segments are there. Whereas in case of prokaryotic, it is very simple, and also it is very easy to define. But in case of eukaryotic gene, it is a highly complex structure. So that we will see now. Okay, now see, if you see here, so it is actually a complex array of regulated sequences that govern the transcription of the gene. So what are all those things? We'll see. So this is actually a structure of a eukaryotic gene. If you see, so here you have got exon is there, intron is there, exon, intron, exon, intron. So likewise, so you have got coding sequences as well as non-coding sequences will be there. So actually exons are present. So exons are actually are called as the coding sequences, whereas introns, if you talk about, they are the non-coding. Now this is the structure of the eukaryotic gene. So we can say, we'll see now what are exons and what are introns. Now coming to exons, now exons are nothing but they are the sequences in the gene that are found in the functional messenger RNA. What are those? They are the sequences, or you can say the gene sequences which are found in the messenger RNA molecule. It includes coding sequence. Just now I have told you it includes, because this color indicates the coding sequence. So it includes a coding sequence that, but may also include non-coding sequence. Sometimes it also includes a non-coding, but major it includes a coding sequence. So the beginning of the first exon defines the site of initiation of transcription, since there is no processing of the 5' end of the primary transcript. See here, this one. So the end of the final exon will define the site of the cleavage, this one here, site of the cleavage of the primary transcript at the 3' terminus. So termination transcription does not terminate at this position, but continues some distance downstream. So what are exons over here? We can say actually in a eukaryotic gene, it is a discontinuous, it consists of a number of discontinuous segments. And these segments actually, because see, it is not a uniform structure where, so it is a discontinuous, it is broken down into, like you have exon, intron, like exon, intron, intron, exon, like this. Okay. So this is how they have given. So the basic structure of the eukaryotic gene, if you observe, so the sequences that are present, the gene sequences that are present in a messenger RNA molecule, you call it as exons. It includes both the coding as well as the non-coding sequences. So the beginning of the first exon actually defines the site of what? Initiation. See here, this is the transcription initiation, whereas at the end it will initiate the what do you say, the terminus, but it will not terminate, it will go on, because this is again followed by initiation. So like this, it is a discontinuous process, or it consists of discontinuous segments like this. Okay. Now coming to introns. Now, what are introns? Now, introns are the intervening sequences in the GNA that are removed in the formation of the functional messenger RNA. So, what are introns? Are nothing but they are the intervening sequences, whereas exons are what? Just now we have told you, they are nothing but the gene sequences which are present on the messenger RNA molecule, whereas introns are the intervening sequences, okay, of the gene, and which are basically removed, okay, during the formation of the messenger RNA. So it includes non-coding sequences. It includes only the non-coding sequences, but the non-coding sequences. So, but sometimes what they are saying is there are instances of the alternative processing where sequences can be both introns and exons. Exons. Now, so with respect to the, this is about the introns. So we will not go into the details. So, but what we can say is the, if you talk about the eukaryotic gene, now eukaryotic gene actually is what? It is a highly complex structure. Now it is a because it consists of a discontinuous segments. So it is highly complex structure, and it consists of major exons as well as the introns. Exons are nothing but the gene sequences which are present on the messenger RNA molecule. It consists of both the what do you say, coding as well as a non-coding sequence, means major coding sequences, but sometimes it can also have the non-coding sequence. But if you talk about with introns, introns actually have the non-coding sequences. Basically it has got the non-coding sequences. Okay. Now next is coming to the viral gene. Now if you talk about the viral gene, now viral gene actually we call the genes which are present in the viruses. So those are also called as nothing but the retrovirus, say for example. So if you talk about the retrovirus, now retrovirus is what? Actually is nothing but an RNA virus. You all know actually the genetic material, if you talk about with reference to prokaryotes and eukaryotes as well as viruses. Now in the prokaryotes, what is the genetic material? It is nothing but the what? DNA or RNA? It is DNA. Whereas with reference to the eukaryotic organism, the genetic material is also DNA. But when you talk about the in plants, it is RNA. Whereas when you talk about the viruses, it has both RNA as well as the DNA. So that is the reason we say, the viruses consist of both RNA as well as the DNA as a genetic material. So based on that, we call it as RNA viruses as well as a DNA viruses. So here they have given an example of the viral gene, that is retrovirus, whereas the retrovirus actually is nothing but an RNA virus. That means here the genetic material is nothing but an RNA. So here a retrovirus is nothing but an RNA virus that is duplicated in a host cell using the reverse transcriptase enzyme. So to produce the DNA from its genome. So the DNA actually is incorporated into this host genome with the help of an enzyme. That is what we call integrase enzyme. So the retrovirus actually is an enveloped virus. It is an enveloped virus that belongs to the viral family called as retroviridae. Now here they have given the structure of the retrovirus. That is, it consists of an enveloped particle about 100 nanometer in diameter. And you all know that virus particle is also called as a virion. So the virion actually possesses what? Two identical single-stranded RNA molecules. Whereas DNA, if you talk about the structure of DNA, it has got two double-stranded. Whereas RNA, you all know it is a single-stranded. But here, in case of virion, it contains two identical single-stranded RNA molecules. Okay. Then next is the envelope. What does it consist of? It consists of the lipids obtained from the host plasma membrane during the budding process as well as a glycoprotein. Okay. Now next is what? RNA. Now RNA consists of what? Uh, it is a dimer RNA. So the RNA, one second.
Okay.
Sorry for the disturbance, students. Okay. Now, next is coming to RNA. Now, RNA, you all know, consists of a dimer RNA, and the RNA also has a non-terminal, terminal non-coding regions. And RNA, you all know, it is important in replication, and it will help in the gene expression. That means it will form the proteins which help in the gene expression. Now, coming to the protein. Now, the protein consists of what? GAGs, that is GAX proteins. Then you have got the protease, Pol protein, and also the envelope proteins. Now, GAG proteins are the major components of the viral capsid, which are about 2000 to 4000 copies. And protease is actually expressed differently in different viruses. So the next is what? How actually these retroviruses multiply, they have given. So when you talk about the multiplication of retroviruses, now the retroviruses actually will integrate their own genome into the germ line. Their genome is passed on to the following generations. So these are endogenous retroviruses, and they are contrasted with exogenous ones. Okay. And when the transcription is going on, the reverse transcriptase, what it will do, it will transcribe the RNA to the DNA. Means you all know, actually, if you know the what is it, dogma of protein synthesis, you all know, actually, from the DNA RNA is being formed, and from the what do you say, proteins are being formed. So like this here, what it will do is, because since these viruses have got the RNA reverse transcriptase enzyme, it will try to transcribe this RNA to DNA. So from the RNA, you form the DNA. So these inserts actually are transcribed by the enzymes of the host, a new RNA molecules that enter into the cytosol or the cytoplasm. So some of these RNA molecules, what happens, are translated into the viral protein. Okay, so like this. So it is important to note that this reverse transcriptase actually is present in its capsid. Otherwise, it is unable to utilize the enzyme of the infected cell. So the transmission, how it actually transmits this virus is from cell to cell, and it can be transmitted through the fluids, and it also can be transmitted through airborne materials. Okay. Now, next is what? The gens. Now, gens is the retrovirus genomes commonly actually contains these three open reading or open reading frames, which are actually called as the gens. Now, they also possess what? The group specific antigen, polymerase, as well as the envelope. Now, these are the three, what do you say, the gens or the genomes, you can say, retrovirus genomes. Now, group specific antigen, polymerase, as well as the envelope. When you talk about the group specific antigen, it codes for core and structural protein. Polymerase will code for reverse transcriptase as well as for the protease and integrates, and the envelope will code for the retroviral code protein. Now, next is what? Your provirus. Now, provirus actually will be incorporated into the host genome as a provirus, and which can be passed on to the progeny cell. Then the retrovirus will be inserted at random into the host genome. So because of this, it can be inserted into the oncogenes. So in this way, some retroviruses can convert the normal cell into the what do you say, the cancer cells. Now, then retrovirus life cycle they have given. So retrovirus life cycle actually is a very, very important aspect. So you all know, actually, the cancer cells as well as the cells, because they all have these retrovirus viruses. These retroviruses actually have got the reverse transcriptase. So because of this reverse transcriptase enzyme, actually today you're not able to actually find any vaccine or produce any vaccine for the cancer cells, as well as for the malaria, as well as even for your what do you say, the what is that, AIDS. So here the retrovirus plays a very, very significant role because of this concept, because retrovirus has got the capacity, actually, you all know, from DNA RNA is formed, from RNA protein is being formed, but in this case, actually, from RNA DNA is being formed. So that is why, actually, because of this concept, or because of this phenomena, today they are not able to actually because you all know, viruses, they are active only inside a host. When inside a host, actually, what will happen, we don't know. So because of the retrovirus mechanism, or retroviruses, actually, they are not able to find out any vaccines till now. So it is a highly complex, the life cycle of these retroviruses is highly complex, because of the presence of this retrovirus, what do you say, the reverse transcriptase enzyme. Okay. So this is, I think, in your syllabus it is not there. We'll not go into the details of this. Just we'll see what are the different types of what do you say, the uh, genes. Okay. Now, this is the structure of the retroviruses they have given. Retroviruses contain open reading frames. Okay. Then you have designated by GEG, PRO, POL, as well as the ENV genes. Just now I've told you which allow for the translation of the precursor protein. Then at the GAG, PRO, POL, and ENV proteins, they are actually being synthesized. They come together to assemble the progeny viron. Then, as the complex increases in size, it will apply the pressure to the membrane, causing the virus to bud outward. Finally, the virion particle actually is pinched off and it is released into the matrix. So, when once it is released into the matrix, that is during and after that, what happens? The GAG precursor is cleavaged by the viral protease enzyme, and the mechanism behind this is you all know. Okay, so this is how. So, when once it is being released, the cleavage will start of all the different types of proteins. So, when once the cleavage starts, a number of virus proteins or the virus particles actually will be formed. They will become mature, and then they will infect. So like this the infection will take place, and the viral life cycle will go on. So that is why actually when you talk about this viral gene, that you should know about actually the what is it, retrovirus, uh, life cycle of the retrovirus, because retrovirus, that is RNA viruses, actually are more dangerous. So RNA viruses actually they are present in the AIDS what do you say, the virus AIDS virus actually is an RNA virus. Okay, so like this you have different types of the diseases in which RNA virus is there. So wherever RNA virus actually is there, so such type of diseases actually, or you can say the infections are not treated. Up to the extent, only up to certain level actually they are treating. Why? Because I told you, because of the presence of the reverse transcriptase enzyme. Okay, so this is the concept, or you can say the life cycle of the RNA viruses. Okay. So now coming to the summary, you all know actually when we talk about the gene and all those things. Now, so what is gene? You all know it is nothing but a unit of heredity, you can say, or it is a functional unit of heredity or a fundamental unit of heredity that is being transferred, or it transfers the characters, you can say, from one generation to another generation. Now, we have seen a brief history of the gene, who were the scientists actually who tried to give the concept of the gene, or who worked to give the structure of a gene, to give the what do you say, the not only the structure but also the chemical composition and all those things. Then next we talked about the fine concept. When you talk about the fine concept, you go into the details of the gene. It has got three regions, I have told you, that is recon, muton, as well as the cystron. Apart from that, we have also talked about the different terms like complon is there, okay, replicon is there, like this. Then we have talked about the different types of what do you say, the genes, that is a promoter gene. Then you have got the, sorry, not promoter, the bacterial gene. Then that is prokaryotic gene, eukaryotic gene, as well as the viral gene. Now bacterial gene, it is present in the bacteria. When you talk about eukaryotic gene, when you talk about the bacterial gene, then you have to talk with reference to the operon concept and all those things, where you have got the structural genes are there, operator gene is there, regulator gene is there. Different types of genes will come into picture when you talk about the bacterial. When you talk about the what do you say, the eukaryotic gene. Now eukaryotic gene is highly the complex. You all know, here we need to go about the details with reference to, because actually you have got the it is a highly complex structure, and it has got a number of coding as well as the non-coding sequences with reference to introns as well as the exons, and it is a discontinuous process. So that is the reason why it is a highly complex structure. Then next we have talked about the viral gene. When you talked about the viral gene, you have to talk about the genes that are present in the viruses. Now you can take best example of the RNA virus, like retrovirus. So what is retrovirus, where actually it is present, and what is the concept or the mechanism of these retroviruses, how the life cycle will take place, what are the genes or what are the various proteins that are present actually in the viruses, and how actually the mechanism or the life cycle of these retroviruses will take place, and how they cause infection when they invade the other foreign organisms. So that is all about the fine concept of gene. Is it okay, students? Hello? Yes? Hello?
Okay, madam.
Okay, so actually this is with reference to the concept of gene. అందరికి అర్థమైందా? Yes or no? Yes.
Yes, ma'am.
Okay, now we will go to the second concept. Now we will talk about the Mendelian principles. Okay, so you all know actually who was Mendel and what are the various principles actually were adopted by the Mendel, and where he worked and on what concept he worked and what are the various laws that actually he has given. Okay, one second. Hello. 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 Hello.
Yes, ma'am.
సారీ మా కొంచెం డిస్టర్బెన్స్ అవుతుంది అందుకనేసి నేను ఏదైతుంది I'll just take. Okay, wait, don't go away, wait.
Okay, sure.
Hello students, hello. Hello. Hello.
Yes, ma'am.
Hello.
We are here, ma'am.
Sorry ma, I'm not able to show share that what do you say, PPT. So I will
Okay.
just tell you later on. I'll share you the PPT. Okay. So this is what
Okay,
the
sure.
same what I told you just now regarding the concept of gene and all those things.
Okay,
So
sure.
what I'll do is we will go to the second chapter. This is what regarding the first chapter is regarding the concept of gene and all those things. I will just explain it to you regarding the second chapter, that is the Mendelian principles and all those things. So you all know actually who was Mendel. Okay. So later on I will send you. Okay.
Okay.
Now coming to coming to the Mendelian principles, that is the second chapter. First chapter was the concept of gene. I think you might have understood very clearly about the concept of gene. That is a fine structure of the gene. Now we will push on to the Mendelian principles. What actually, who was Mendel? What actually he has given? Okay, so you all know actually Gregor Johann Mendel was the pioneer, and he is actually considered as the father of genetics. You all know, and he was actually born in what is a very poor family, that is a farmer's family. And he was very much from his young days, he was very much interested actually in farming. Along with his parents, he used to go. So he's actually how actually Mendel became famous is when he was going along with his parents and all those things. When he was very small, when he was going when their parents were going for farming, he used to observe the plants, how they are growing and all those things. So like that he developed an interest, and then he pushed on to actually the going for the crossing of the plants and all those things. So most of the experiments actually, his experiments basically, or his it has started from the pea plant, as you all know. So he did the experiments, crossing experiments, you all know, from what is it, the pea plant. And what he did was actually he selected 7 characters, you all know. So the Pisum sativum actually is the scientific name for the pea plant. So the pea plant actually, what he tried to do is when he selected this pea plant, he could observe a number of characters among this pea plant. So he thought, actually, why not we take this particular plant and go and cross I mean, the pea plant, taking different characters. So like that, what he did, when he took only one character, he called it as a monohybrid cross. When he took 2 characters, so like this, he actually tried to study 7 characters. So 7 contrasting features, or you can say, of the what do you say, the pea plant. Actually, he has taken, selected, and he has worked on it. And various, like you have, maybe with reference to height, like tall and dwarf, or the color of the cotyledons, or the color of the, I mean, the shape of the seed, like they may be round or wrinkled, or the seed coat, pods. So like this, he has taken a number of what do you say, different contrasting characters. About 7 characters actually he has taken, and each time he tried to actually take different character and study different character, and actually he has put them, he has crossed them, and he has given us the, in the form of those laws. So those laws today we call it as nothing but the Mendel's laws. So you all know actually the different types of Mendel's laws are nothing but the law of dominance, law of segregation, as well as the law of independent assortment. Now, when we talk about the law of dominant is what you all know. Actually, what do you mean by the law of dominance? That actually the dominant character actually will be present in the F1 generation. So before we go to the all these laws and all those things, you know, what is a dominant character, what is a recessive character, what do you mean by phenotype, what do you mean by genotype, what is an allele, what do you mean by allelic frequency, and all those things. So what he tried to do is when he crossed, he could observe that certain characters actually try to remain dominant, means it tried to occur generation after generation, they were occurring. So such characters he called them as a dominant characters. And certain characters actually were not able to see, he was not able to see it in a cross, so they skipped the generations. So such characters he called them as the what do you say, the recessive characters. So we can say dominant characters occur in all generations, or they are present, they are seen in every generation, whereas the recessive characters are not seen in all the generations, so they skip the generations. So like this, he called it as a dominant character, then he called it as a recessive character. Then he talked about the phenotypic and genotypic ratio. Now phenotypic ratio is nothing but, I mean, the phenotype is nothing but an external what do you say, morphology, whereas the genotypic is, it talks with the genes, that is the number of genes that are, or you can say the genetic makeup of an organism, you call it as nothing but a genotype. So like this, Mendel, what he tried to do, he tried to give different what do you say, the phenomena or the concept or the terminology for the different. Then when he crossed, he gave the genotypic ratio, phenotypic ratio, then allele. What do you mean by allele? Allele is nothing but actually every gene has got a pair of alleles. So these two alleles together will constitute a gene, means one gene consists of a pair of alleles. So these alleles actually are responsible for the transmission of characters. That is, the gene is responsible for them. Okay, so like this, now the different 7 characters are already I have told you, with reference to the seed structure or the form, cotyledon structure, then the flower color and the form, then the pod color, and then the stem color. So like this he has taken. Okay, so now coming to the cross between the pea plants differing in a single pair of contrasting characters, you call it as a monohybrid cross. So when he crossed only one, when he took only one character, say for example tallness and dwarfness, he called it as a monohybrid cross. Then what he did later on, he tried to take the color of the seed coat. Means he studied 2 contrasting characters at a time. Then he called it as a monohybrid cross. So based on this phenomena, what he gave, based on the phenomena of dominance, actually he gave the monohybrid cross. So for Mendel's monohybrid cross, actually how it came is when he tried to cross a homozygous tall plant with a homozygous dwarf plant, what he could observe is he could observe that the F1 hybrids were all tall, thus showing that the complete dominance of the tall over dwarf. And when these F1 hybrids were allowed to self-fertilize, again they produce both tall and the dwarf plant in the F2, but in the ratio of what, 3:1. So the appearance of dwarf in the F2 generation indicate that the process of segregation. So based on this monohybrid cross, what he did is he tried to actually give the law of segregation. So like this, Mendel has given three laws. One is called as the law of dominant, then law of the segregation, and lastly the law of independent assortment. And the law of independent assortment actually is based on Mendel's dihybrid cross. Mendel's dihybrid cross. So like this, that when he tried to actually give the dihybrid cross, then what he did is law of independent assortment came. So in the dihybrid cross, what he tried to do is he tried to actually take two characters. Say for example, he has taken like a color as well as the shape of the seed, like yellow round seed. When he tried to cross the yellow round seed with the green wrinkle seed, say for example, then he observed that in the F1 hybrids all had yellow round seeds. They all had the yellow round seeds because yellow is a dominant character, round is also a dominant character when compared with the green and wrinkle seeds. So when these F1 hybrids were allowed to cross among themselves, then what happened? They tried to produce four types of seeds in the ratio of 9 is to 3 is to 1. Okay, 9 is to 3 is to 3 is to 1. That is what we call as a phenotypic ratio. So, yellow round 9, yellow wrinkle 3, green round 3, green wrinkle 1, like that. Okay. So, besides getting the ratio of 3 is to 1, the monohybrid cross, what Mendel got is the ratio of 9 is to 3 is to 3 is to 1. So, by stating this, what he told, explained by Mendel, he stated that when the actually, so based on these phenomena, dihybrid cross, he gave the law of independent assortment. So what he tried to explain is, so when the parents differ from each other in two or more contrasting characters or factors, that means when the parents differ, okay, from each other in two or more pair of contrasting characters or factors, then the inheritance of one pair of character is independent to that of the other pair of character. So such a law, he called it as the law of independent assortment. So these are nothing but these are the three laws actually that Mendel has framed, okay, based on the his cross. So who is Mendel? You all know he is nothing but the father of genetics. What he did, he studied actually the pea plant, and he took 7 characters of this pea plant, that is pisum sativum we say. Okay, and then what he tried to do, he observed when he tried to cross this pea plant taking different characters, then he could observe a lot of changes were occurring. So first he took only one character at a time, then he studied 2 characters at a time, then he studied 3 characters, like this. Based on this, Mendel framed 3 laws. What are those laws? One is called as the law of dominance, then law of independent segregation, and the law of independent assortment. So Mendel's monohybrid cross means law of segregation is based on Mendel's monohybrid cross, whereas law of independent assortment is based on Mendel's what dihybrid cross. So you all know what is monohybrid cross and what is the dihybrid cross. So after that, what he tried to do, he tried to actually work on the trihybrid cross. Now, so 3 characters at a time. So when Mendel crossed 3 pairs of contrasting characters, he called it as a trihybrid cross. For example, a plant height. Say for example, that is either tall or dwarf, or when you take a seed shape, whether it may be round or wrinkled, or a seed color, yellow or green. So that means when you're studying 3 contrasting characters at a time, then you call it as a trihybrid cross. So the F1 generation of these trihybrid cross actually are heterozygous for all the 3 traits, and they produce 8 type of gametes. So when they self-fertilize, then what happens? These gametes will have an equal chance of giving rise to 64 different combinations. Okay, so in this, what happens is when there are more than 2 pair of contrasting characters, what we do actually, Mendel's when he tried to explain the monohybrid, hybrid, whatever, he tried to actually explain it with the help of a Punnett square. So you all know Punnett square actually is nothing but it is a widely accepted what do you say, a tool to explain. So it is actually based on rows and columns. So here the fork line method we can say, it is used instead of sometimes what is it, instead of a Punnett square, some of the people also use the fork line method. But if we use a Punnett square actually will give you a clear cut picture that instead of crossing it, the same gametes and the same offsprings whatever actually are obtained based on the F1 generation, if you put them in the form of a square and then try to cross it, then it will give you a clear cut what do you say, idea about the different generations like F1 generation, F2 generation, and F3 generation. So apart from this Punnett square, actually some scientists also use the fork line method actually to identify or to predict the genotypic as well as the phenotypic ratio. So in one way, this Punnett square as well as this fork line method will help you to predict. See, you all know actually Mendel's laws are based on probability. So based on probability only, he has given us the ratios, whether it is a phenotypic ratio or whether it is a genotypic ratio. So like that, he has actually tried to cross, and he has actually put them or tried to interpret the result or try to predict the result by putting them in a Punnett square or by putting them in a four-line method, and then give us the different ratios. So when with reference to the tri-hybrid cross, when he did this, about 64 different combinations actually he got it. I mean different types genotypes in the F2 generation. Like the phenotypic ratio actually was in the form of 27 is to 9 is to 9 is to 3 is to 3 is to 3 is to 1. So like this he got the about 64 different combinations of the genotypes in the F1 generation, and 9 you can say different types of phenotypic ratio. So like this Mendel tried to actually study the three different crosses, and he got the phenotypic as well as the genotypic ratio. Okay, so this is about the monohybrid cross, dihybrid cross, as well as a trihybrid cross. Now coming to the back cross as well as a test cross. So what do you mean by back cross and a test cross? Now when the F1 individuals are crossed with the one of the two parents, then you can call it as a back cross. You can call it, for example, in such when F1 is back cross, okay, to the parent with a dominant character, okay, where no recessive individuals are involved, so then you call it as a dominant back cross. On the other hand, when it is crossed with a recessive parent, then what happens? Then you call it as a recessive back cross. So in case of this the back cross, so back cross means what? When the F1 individuals that are being produced, you can say, or when the F1 progeny actually is being crossed with one of the two parent, okay, then such a cross you call it as a back cross. So in such crosses what happens is, if you are crossing with a parent, suppose a parent is having a dominant character, then what happens? No recessive individuals will be obtained. Suppose if the parent is a recessive, then both the phenotypes will appear in the progeny. So when with both of these crosses, well, both of these crosses are back crosses, only the cross with the recessive parent actually, and the cross with the recessive parent actually is no, you get it. Okay, now this is about the back cross. Now what do you mean by test cross? Now test cross is nothing but when the F1 individuals are crossed with the recessive, okay, parent, then you call it as a test cross. You call it as a test cross, because actually this test cross will help you to find out whether an individual is homozygous or heterozygous. Okay, so it will help you whether it is homozygous or the heterozygous. So this is about the back cross as well as the test cross. So apart from the monohybrid cross, dihybrid cross, as well as the trihybrid cross, Mendel also tried to actually apply the back cross as well as the test cross. So back cross, when he did, it is nothing but a crossing between the F1 individuals with either of the parent. Okay, it may be a dominant or it may be a recessive, then you call it as a back cross. But when you are crossing the F1 hybrid individuals with a recessive parent, then you call it as a test cross. Then you call it as a test cross. So this is all about the Mendel's what do you say, the different ratios that they have given. Okay, so now you all know. So what are Mendel's laws? Laws. So Mendel actually has given three laws to us. So how he has actually selected the plant, and what are the different seven pairs of contrasting characters he had taken, how he crossed them, and based on the crossing and based on the probability concept, he has given us the three laws. That is the law of law of dominance, law of segregation, as well as the law of independent assortment. So Mendel's three laws, you all know. So law of dominance, law of independent segregation, and the law of independent assortment. Okay. Now coming to, so apart from this, so how actually the success? Now, what is how Mendel actually gained success? What factors actually led to the success of these Mendel's experiments? So why? Because you all know actually the Mendel success was dependent on his careful selection of the pea plant, because he has selected the pea plant, pea plant, and he has very keenly observed the pea plant, and also he has selected the seven pairs of contrasting characters very keenly he has observed. So he was very lucky because the plant did not show any incomplete dominance at all or any epistasis. So that is the reason why he was successful in doing it. So whatever the plant that he has selected, that is the Pisum sativum or the pea plant, actually was what this plant did not show any incomplete dominance. You all know what do you mean by incomplete dominance? Incomplete dominance means not a single factor will be dominant. Okay, so both the factors actually here will play a significant role. So that is the reason why actually both of them will play a role, and so it is called as an incomplete dominance, where a single character actually is not completely dominant. Both the characters actually will come into the picture. When do you call a dominant character? When actually when do you call a dominant character? When the trait, or you can say the character, which mask or suppresses, okay, the other character, then that character you call it as a dominant character. That character you call it as a dominant character. So in case of Mendel, what happened is, when he selected the pea plant, this pea plant did not show any incomplete dominance. Means whatever the character that he has observed, 7 pairs of contrasting characters he has observed, you all know. So there he did not get the incomplete dominance phenomena. Incomplete dominance means where the character, okay, I mean, did not actually show a complete dominance. It, okay, I mean, did not show the incomplete dominance. Means both the characters will play a role. So here the characters played a significant role. Why? Because it showed a dominant character over the other character. So that is why he was successful. Then second aspect is what he studied, all the 7 pairs of actually different characters he has studied individually in both the parent as well as in the hybrid. So what he did is he classified the offsprings according to their characters as dominant character, recessive character, all those things. So he tried to maintain the records of these having the particular characters. So this also actually helped Mendel in one way to get the success. Then the second thing, the third aspect is what Mendel studied the inheritance of one character at a time. He did not mix up, he did not make a mess of it, because what he tried to do is one character at a time, one character at a time he tried to study. So that is the reason why also he actually he got the success. So he studied the inheritance of one character at a time, whereas his predecessors, what they try to do, they consider the whole organism as a whole. So that is the reason why Mendel was successful actually in getting what do you say to the success of his experiments. And he carried out the experiments till F1 and F2 and F3 generations. Okay, he carried out experiments to F2 and F3 generations also. So he did not consider as a whole animal, but rather he studied a single character at a time, and he has gone up to F3 generations. Still three generations actually he tried to I mean cross it, and he tried to give us the genotypic as well as the phenotypic ratio. So these concept actually led Mendel to become a what do you say to become successful in his experiments. So this actually made Lendl, I mean, not only he got the fame, but at the same time, he got the success for his experiments, and he was awarded, you all know, or called as the father of genetics. So that is how actually the Mendel tried to gain or achieve the success. Okay. Now other things, other aspects is actually he has just now I've told you, actually he became when once he was successful in doing all these things and all those things. So Mendel not only try to give I mean, Mendel's, you all know, actually experiments. I mean, only few records he has kept. He has not kept the records to the fullest extent. So in that case, what happened? What happened is his experiments did not, I mean, come into the to the society when he was alive. Only after he died, actually his experiments again were proven by different scientists, and they try to actually prove that his experiments, whatever the experiments he has conducted, were true. And that's how actually he became famous after his death, but not when he was alive. So that is that thing we should note it. And this is how actually the Mendel became the famous. So these are the three, we can say, the various principles actually that Mendel has given, and which made him success. And also because of his experiments, he was being conferred the title as the father of genetics, you all know. Okay, so this is about the Mendelian's inheritance. Okay, or you can say the Mendel's principles or the Mendel's laws. Okay, so what are the three Mendel's laws? You all know that is law of dominance, law of and, independent law of segregation, then law of independent assortment. I mean, the category will be first law of dominance, then second category will be the law of independent segregation, and lastly will be the law of independent assortment. Because what he tried to do, first he crossed monohybrid. He took single character, then double character, then three characters. So like this he went on. So based on the what is a monohybrid cross, he actually gave the law of the what do you say, segregation, and based on the dihybrid cross he gave us the law of independent assortment. So that is how actually he could succeed in doing this. Yes, students. Hello. Hello. Hello.
Yes, ma'am.
Are you able to follow?
Yes, ma'am.
Okay. So I think sorry for I mean not I mean showing you the PPT. So tomorrow I'll show you. Otherwise, I'll put it in the group. Okay. So now we
Okay.
have actually talked about what the concept of gene, and we have talked about the Mendelian principles, or you can say the Mendel's
Yeah.
laws. Mendel's laws. Okay. If you have any doubt, if you have any queries, you can ask. Yes. Any doubt you have? Yes. Hello. Nobody is having any doubt.
No, ma'am. It's clear.
Is it clear? Okay. The
Yes,
concept
ma'am.
of gene. First, we have talked about the concept of gene or the fine structure of gene. They mean both mean one and the same. Okay, so don't get confused. Concept of gene or fine structure of gene. Here you have to talk about the gene, history of gene, actually who were the scientists who were involved. Then talk about the structure of the gene, that is concept or the three different types of genes. Especially, what are those three different? I mean, the three regions of a gene. Okay, first you talk about that is recon, muton, and ceston. Then talk about the different types of the genes. Okay, eukaryotic gene, prokaryotic gene, as well as the viral gene. Now, when you're talking about the Mendelian principles, now in Mendelian principles, first here you have to talk with reference to Mendel. Who was Mendel? Okay, a brief history about the Mendel. How actually Mendel got interested in actually going about with the his laws, because actually he was born in a farmer's family. So since he used to go along with his parents to the farm, he used to observe very keenly when the plants were growing and all those things. So he developed that interest, and later on when he was studying and all those things, he selected the plant, not the animal. So he selected the plant. So that too, he selected a pea plant. Why he selected the pea plant? Because he was a keen observer, and he knew how to go about with all those things. So that is the reason why, and he selected this pea plant. So that I mean the pea plant selection of this pea plant led him success. Just now I mentioned, because the pea plant actually never showed the incomplete dominance. So, and he studied though he studied 7 characters, but he studied one character at a time. He did not make a mess. As a whole organism, he did not do it one character after one another. Like that, he tried to study, he tried to actually cross it, and up to 3 generations he studied, and he tried to give the both the phenotypic as well as the genotypic ratio. So that is the reason why he was very, very successful in doing so. So because of that reason, actually he became very, very famous. But the sad thing about him was, when he was alive, he could not become fame. Why? Because what do you say, the works that Mendel actually tried to do, they were they did not come into the picture, because he did not maintain the records and all those things. Only very few the records he maintained, and on those, based on those records only, later on the other scientists, what they try to do, they try to work on it, and then they try to prove, yes, what Mendel actually has given the segregate. But of course certain loopholes are also there, and they try to actually try to modify those Mendel's laws, that we call it as nothing but extra what do you say, or you can say non-Mendelian inheritance. So that we'll talk it in the next class. So today, now what I talked is about the Mendelian inheritance based on his 3 laws. That is, these 3 laws are very, very important. That is law of dominance, law of segregation, and the law of independent assortment. So how he has given, you all know. So before going to all these things, first you should know what do you mean by dominance, what is a dominant character, what is a recessive character, okay, what do you mean by F1 generation, F2 generation, F3 generation, what is a phenotype, what is a genotype, how Mendel took it and how he crossed. So what is a monohybrid cross, what is a dihybrid cross, okay, what is a trihybrid cross. So like this he tried. And what do you mean by Punnett square? Because when he was crossing, to make the society know about all these things, he had to actually put them in a very simplest form. So what he tried to do, he tried to put them in a cross, and also he tried to use a Punnett square. Punnett square is nothing but a square where actually it will give you a clear cut idea. I mean, if you put the F1 progeny into that, put it and then try to cross it, you will get a, I mean, you can easily understand, and at the same time, you can also segregate them easily and try to get the phenotypic as well as the genotypic ratio. So that is how not only he used the Punnett square, it is nothing but a form of, in the form of a square. So actually why it is called as a Punnett square? Because it is given by the scientist actually, or you can designed by a scientist, you can say, named as Punnett. So that is why it is called as a Punnett square. So he also used the fork line. So like these different methods are there, but these two are actually are very very important, which have been used by Mendel very often. So that is the reason why these two what do you say methods have gained a lot of significance, which because it made them easier to understand to the common people. So that is why the Punnett square as well as your the forked line method has gained a lot of significance, because it is easier for you. You can just put the genome, I mean the what do you say, the gametes, and then you can try to cross it, and easily you can try to predict the genotypic as well as the phenotypic ratio. So that is the reason why actually he used both those things to make the what do you say, the audience also easier, so that they can very easily understand his concept. So that is how it has come. Okay. Yes, is it okay, students? Hello.
Yes, ma'am.
Hello. Okay. So tomorrow, sorry for the not sending it, because and also wasted a little bit of time. So tomorrow we will go to the next chapter. Okay.
Okay, ma'am.
Okay. Now it's time. Now we will leave.
Teacher, madam.